This video will demonstrate the use of brain atlases in BESA Research. We will have a look at brain activity evoked by a visual motion field in an MEG experiment. We have the average data available here, and have co-registered the dataset with an individual structural MRI of the subject. I first define an interval in the data segment that I want to analyze, which is the motion stimulus here with 129 averages, and then right-click to define my choice of subsegment and filters for source analysis. This brings up a small dialog. I will adjust the time to include -100 to +250 milliseconds relative to the stimulus. Then I will focus on a filter set from 3 Hz to 40 Hz, which should bring out the evoked activity nicely. Now I start the source analysis module and maximize the window. I can see the global field power here, with 2 peaks for the primary visual processing and the motion processing, and on the left a butterfly plot of all channels and the PCA. I will open a predefined solution for this dataset that shows the activation of primary and subsequent visual processing areas well. The first 2 dipoles model the primary visual activity. Dipoles number 3 and 4, the green and purple ones, model the lateralized activation that is due to the motion. You can see how their activity aligns with the second peak of the global field power. This data peak is not apparent in a static stimulus. Let us look at the anatomy for these motion-related sources. I can press the A key on the keyboard to bring up the anatomical view with the MRI of the subject. Let me switch to a display of 2x2 sub-windows with 3 MRI slices and a 3D head sliced at the currently selected source. I can maximize the window here. I will now add the Atlas overlay by clicking on this icon here in the toolbar of the 3D window. By default, the atlas that is selected is the Brainatom atlas. I can see that atlas name here at the right. The display shows the Brainatom area for the current crosshair, as well as the Talarak coordinate. I can change the blending mode from the contour mode that is currently shown to a color coding by right-clicking and changing the Atlas blending mode. 2 different types of color coding are available. The brain color one tries to assign similar brain areas similar colors. It is apparent that the lateral dipoles both are situated in the V5 MT areas. It is very easy to switch between the brain atlases. Just right-click and select Options. Then you can toggle through the different atlases. The other ones available are AAL, Brodmann, AAL 2015, and Talarak. Let us choose Brodmann for the moment. You can see that the area changes here. How about overlaying atlases with another imaging modality? For example, if my experiment has fMRI data. I can load my fMRI data using the Image menu. For this data file, I have a map computed with Brain Voyager already available. So I can choose this map and then wait for the configuration dialog to appear. I can apply a voxel threshold criterion here to only show the significant clusters. Another option is to use the degrees of freedom and work with the p-value. Anyhow, I will choose 50 for the moment. Then I can see the statistical map overlaid. Let us go back to the options to change the opacity value of the Atlas overlay. If I take it right down, then the fMRI map dominates. If I choose a value between 20% and 30%, then I can see the image as well as the atlas regions overlaid on the brain quite nicely. If you would like to use any of the brain atlases in our publications, please remember to quote the author of the brain atlases. The BESA help provides all the references for the different atlases implemented. And to the brain atlas topic. Here you see the description of all available atlases and the references for these. Thank you.